Hello, this is Tutor Do, and today I'm going to show you how to do a genetics problem. Talking about non-recombinant offspring when you're given the parents, and then you have to find the F1 and the F2. So the problem is wild-type humbugs have brown bodies and brown eyes and are not spotted. You have isolated mutations in 3 new autosomal humbug genes. The mutation SP gives a dominant phenotype. So SP is dominant phenotype of spotted bodies. Mutation GR gives the recessive phenotype of green bodies. GR recessive for green. And mutation BL gives a recessive phenotype of black eyes. So BL equals recessive for black eyes. So we have SP for dominant. So that means that SP plus is the recessive, GR plus is the dominant, and BL plus is the dominant. So they say that you cross 2 true breeding mutant strains to produce F1 females heterozygous for SP, GR, and BL. These F1 females are then test crossed to a true breeding, non-spotted, black-eyed, green-bodied male. The phenotypes of 3,000 progeny were scored as shown below. So then they give us a little data table. So the important thing is that you cross 2 true breeding mutant strains to produce F1 females who are heterozygous. So that would mean that they're homozygous for all their genes normally. But the only way you could get heterozygous in this case is if the parents are they're homozygous for all the genes that they have, but they're not all the same type. Some of them have a dominant or recessive, and they're not all the same. So the table that they give us looks something like this. Since they're being test crossed, you don't have to put every single gene because the other gene's always going to be recessive. So I'm just writing out the table that they gave us. Okay, SP plus BLGR, SP plus BL plus GR plus, and SP BL plus GR plus. And we had 4 of this, 1347 of this one, 53, 1390, 2, 74, 61, and 70. So since we're given this, we can make a few assumptions here. Look how big these numbers are. This one and this one. That means that these are the parentals. That's how I came to this conclusion, because they are homozygous for one and a different homozygous for the other, which would mean that these two are the greatest, because that's always what it is with these fly examples. That the greatest number that you see is going to be the genotype of the parents. So now they want us to map the genes, find the distance between the three. So if you look at the smallest amount, which are these ones, look at the smallest two, this one and this one. You look at the parents, you see that the one that changes between them is SP. Because when GR is positive, SP is little, or it's not positive. When GR is not positive, SP is positive. And BL always reflects SP. So, SP is in the middle. And there's no way of knowing which way that the gene was, because it's a three-dimensional matter object. So you have BL and GR. So to find the numbers between them is pretty simple. I mean, they make it really hard make it sound really hard when they're giving you the problems. So between BL and SP, you look at the parents. So they're either both positive or both negative. So you do not count the parents for these, because you're looking for non-recombinant children. So, like I said, SP and BL are either both positive or they're both negative. So 53, we won't count that. We'll count 4. We'll count 74, and we'll count 70. And you divide it by 3,000, because that's how many babies, and you times it by 100. Now you're going to do SP to GR. They're always opposite. So SP is positive, GR is negative, SP is negative, GR is positive. So if they're the same, then you can count them. So we're going to count 53 this time. Oh, we also have 2 here. 53, 4, 2, 61, and we're gonna count, yeah, that's it. So you divide this by 3,000, and you times it by 100. So let's do that math right now. So we have 4 plus 74 plus 70 plus 2 divided by 3,000 times 100. That should give us 5 map units. The map unit is just the unit they use to measure distances between genes. Now we'll do SP to GR. So we have 53 plus 4 plus 2 plus 61. What's that? Divided by 3,000 times 100 will give us 4 map units. So now you do BL to GR, the whole distance between these two genes, and they're either positive, negative, and negative, positive. So you only count them if they're the same. Once again, you don't count the parents, because we're trying to find non-recombinant children. So we are going to count 53. We are going to count 74. We're going to count 61. And we're going to count 70. Now this is where it gets weird. You count both of the double recombinant, which is the two smallest, twice. Because when they were first doing these tests, they found that if you add them both twice, you'll get the actual map unit size between these genes, because you're always a bit short if you don't add these two twice. So you do that, and you divide it by 3,000 times by 100, and in theory, you should be getting 9 map units. So that about sums it up for this problem. You should get 9 MU total, because it's 5 plus 4 for the map distance. So yeah, I hope this helps you. Don't forget to like and subscribe. I'm Tutor. Have a good day.